Hi, everyone. Those of you checking out our newsletter and want to see what kind of information you get from X-ray crystallography, I'm Bob Pike, the chair of the department and the main user of the X-ray, and so I want to show you what sorts of things we can get out of here. So what we've got is, what I'm going to open is a structure that we solved using our new instrument. This program viewer is called Mercury, and it projects the results of the crystal structure. And you can see that I've got a couple of molecules that I can rotate in any direction I want. And what should be interesting first to you is that we have two molecules that look essentially identical. And in fact, let me turn off the hydrogens here. The hydrogens are in white and they make things a little complicated, so I can just shut those off. You recognize some six-membered rings, phenyls, if you remember your undergrad days. And these two structures, I think you'll see, are not quite identical. If I label the atoms, one of them is based on yttrium-1, and here's yttrium-2, and the oxygens over here I've got 1, 2, and 3, and here I got 4, 4, and 4. Isn't that interesting? So there's a couple things going on with these molecules. First of all, there are two chemically separate molecules in this unit cell, in this in the basis of this crystal structure, and the two molecules are almost identical, but not quite. So, for example, let's look at the bond angle between yttrium, oxygen, and phosphorus. Here I see 169.5. Here I see 167, I guess, and then this one is yet different. Let me turn off the label so you can see them better. 164.5. So they're all quite different, at least a little bit different. And then over in this other molecule, I only need to pick one of them, because since we have oxygen 4 every time, any one I pick will be exactly the same as the others because they are created by symmetry. They're all the same. There's a threefold rotation axis in this second molecule, the one with yttrium-2, which means only one of these yttrium, oxygen, phosphorus with the phenyl groups is real. The other two are ghosts. They're generated by symmetry. And the same is true of the NCS, the thiocyanate. Each of them is based on N4, whereas over here you see we have N1, N2, and N3. So in this molecule, in this crystal structure, we have one fully independent molecule right here. Get rid of those measurements. And one 1/3 independent molecule here, where only a third of the molecule is actually unique and the rest is generated by a rotation axis. So that's kind of cool, I think, and that's going to vary from structure to structure. A lot of times you have only a single unique molecule. Sometimes you've got two of them, or in this case 1 1/3, which is a little unusual. Let me turn off the labels and show some of the stuff that can happen. I can project these atoms differently. I can project them as ball and stick. You can see those phenyl rings rather nicely now. They're nice and planar. So these atoms, for instance, form a very nice plane. I'm still picking angles, apparently. Let's clear the measurements and just go to pick atoms. And these thiocyanates look like they're pretty linear. In fact, let me go back to measure angles, and we can see that they're pretty close to 180 degrees around the central carbon. So I can project it this way, but I can project it other ways as well. Here's a really cool one. I can show it as space filling. And now I see how these molecules occupy space. And let me further expand this by showing the packing diagram. The packing diagram shows me the unit cell, that is the smallest crystallographic repeat unit, the smallest unit in space that only has to be repeated in any given direction to completely fill space and create the entire crystal. It almost looks like there's some pieces missing here, but that's just because there's other molecules that are peeking into the unit cell in those spaces. So I can fill it further by adding some more molecules. And you can see that the old adage about nature abhorring a vacuum is really very true. It looks like I've missed a little bit of addition over here. And maybe there. There we go. That nature certainly does not like a vacuum, and will do her very best to fill all of the unoccupied space. You can also see something rather beautiful here with these space filling models. Look at one of the phenyl rings, for instance, this one, where I'll pick out the 6 atoms. Hard to get this one. There it is. You see how it looks like a donut here, where each of the atoms is interpenetrated, is interdigitated with the one next to it. That shows you something about chemical bonding, that when atoms covalently bond, as these carbons do, they help, they actually share electrons, and so their electron clouds interpenetrate with one another. I could turn on the hydrogens again, and that will show that this phenomenon even more, because look how the hydrogen here that I'm pointing out is really part and parcel of this carbon atom. It's almost sunk right into it. All right, let me turn off space filling here and go back to the cap sticks that I started with. And you can still see that the unit cell is pretty complicated, but the unit cell has lots of symmetry operations in it, such as rotation axes and maybe mirror planes and so on. So that those symmetry operations allow us to generate one molecule from another. And so we really only, if we turn off the packing, we only need these 2 molecules to describe the entire crystal structure. Something else you might have noticed is over here. You see how I've got, right here I've got yellow is sulfur. I've apparently got 2 positions for this sulfur atom. And so if I turn on the labels, it'll call this sulfur 2A and 2B. This is called crystallographic disorder. It doesn't mean that the atom is bouncing back and forth between these 2 positions. It just means that it is, as we go from unit cell to unit cell, sometimes it's in one position and sometimes it's in another. And the software can actually figure out the distribution. Is it 50-50 or is it 95-5? That is something we can do. So you can see there's an awful lot we can do with this program. We can learn about distances, both bonded and non-bonded distances. We can see that, for instance, these phenyl rings look like they're interacting. They can sort of stack together. We see that right there. We can measure hydrogen bonding interactions. There's none in this particular molecule, but we might ask, okay, what are the closest interactions? And look, this sulfur is obviously a little bit involved with this hydrogen over here. So if I click on that, it'll expand that molecule. And now we see that there is a fairly close interaction between those 2. And if I dare, let me label these 2. So we'll just pick this one and this one and hit labels so that we can keep track of them. There they are, sulfur 4 and hydrogen 9. Going to turn off the short contact. We know those 2 kind of like each other. Let us go back to the space filling model and see if we can see how they're interacting. So I'm going to go back to space fill. It only gave me these 2, but in a way that's maybe a good thing, because we can see that through this sort of pseudo-hydrogen bonding interaction, the 2 spheres are just about touching. And so there's a little bit of chemical interaction there. It would be increased if the 2 were actually interpenetrating one another any further. All right. So that's some of what we can get out of X-ray. And really, there's a good deal more. But you can see that just the very fact that you can see a molecule in what is essentially a 3-dimensionally rotatable photograph is a really powerful tool. We know that these molecules are real entities. We see their behavior in space. And remember, this is not some sort of modeling. This is not some sort of computational modeling. This actually is an experimental result that came from making this compound, growing a crystal of it, finding how the X-rays diffract from that crystal and reconstructing the atomic positions from that X-ray diffraction map. So anyway, I hope you found this helpful, and thanks for reading our newsletter and being interested in the forefront chemistry we're doing here at William & Mary.